Today, we are diving into something truly revolutionary in the world of biology. We're talking about a new scientific technique that's letting us, for the first time, eavesdrop on the secret conversations happening between the cells in our own bodies. And this isn't just a small step forward. It's like we've suddenly been handed a key to a language we barely knew existed.
Now, I know what you're thinking. That sounds like something straight out of science fiction, right? But believe it or not, your body's doing this constantly. It's firing off these microscopic messages between cells nonstop. And only now are we building the tools to actually intercept and read those messages with incredible clarity.
So, what are these secret messengers? Well, they're called extracellular vesicles, or EVs for short. And you can think of them as the unsung heroes of our biology. They're absolutely central to how our bodies work.
Okay, so picture this. Every single moment, billions of your cells are launching these tiny microscopic packages. Each one is stuffed with vital cargo, things like proteins and genetic material, and it's all addressed to a very specific target cell. That's exactly what EVs do. They are the body's own super efficient internal mail system.
And this is where it gets really, really important. These messages don't just keep us healthy. They're also deeply involved when things go wrong. If we can intercept these packages, we could potentially spot diseases like cancer long before any physical symptoms show up. I mean, they're a literal goldmine of diagnostic info if we can just figure out how to access it.
But, and it's a huge but, there's been a massive problem. For years, trying to study one specific type of EV has been like trying to find one specific needle in a haystack the size of a mountain. And here's why the challenge is just so immense. We're on the hunt for signals on these EVs that are thousands of times smaller than a cell. They also carry way, way fewer proteins on their surface. So the signals we need are incredibly faint. And to make it worse, they're completely drowned out by a huge amount of background noise and all sorts of other look-alike particles.
You know, for the longest time, our tools just weren't up to the job. We could see the vesicles, sure. We could even count them, but we couldn't reliably tell them apart or pick out the rare, super important ones that might be carrying disease markers. The signals were just too weak. We were stuck.
But that is all starting to change. A brand new approach has come along that gives scientists two critical new superpowers, letting them finally cut through all of that noise. The technique is called single sEV-ICP-TOF-MS. Yeah, I know, it's a mouthful, but basically it's a special type of analysis that can look at single particles. And what it does is both simple and absolutely brilliant. It solves the two biggest problems by making the signal way brighter and the background much, much quieter. So here's how they do it. First, they created much, much brighter labels. Instead of the old tags, they used nanoparticles packed with metal atoms. This makes each EV light up like a powerful beacon. And second, they developed this really ingenious filter. See, these nanoparticles have a unique isotopic fingerprint, so the machine can instantly tell the difference between a real EV and random background noise, and then it just throws the noise away. Okay, so that's the theory, but did it actually work in practice? Well, the results, they're pretty incredible. Just to remind you of how tough this problem is, this quote from the research paper points out that we're looking for something with about 10,000 times less signal, fewer of those surface markers than a normal cell. That's the mountain they had to climb. And this chart really shows the power of that first superpower, the brighter labels. Using the exact same sample, the old method could only detect about 17,000 EV events, but with the new nanoparticle labels, that number jumped to over 240,000. That's a 14-fold increase in the number of EVs they could actually see. All of a sudden, vesicles that were completely invisible before were popping up, clear as day. But look, just turning up the brightness isn't enough, because that can also turn up the noise. You have to be sure that what you're seeing is the real deal, and this is where that smart filtering becomes absolutely critical. And the result of that filtering, get this, by weeding out the noise with that unique isotopic fingerprint, the researchers improved their detection sensitivity more than 36 times over. That means they can now reliably find tiny populations of EVs that were completely undetectable before. This isn't just turning up the volume, it's like perfectly tuning to the right radio frequency. So what happens when you can suddenly see things this clearly? Well, this newfound power didn't just confirm what scientists already suspected,
It revealed something entirely new about the diversity of these messengers.
With all that noise gone and the signal boosted, they looked closely at a population of EVs that were known to carry two common protein markers. Before this new technique, this whole group just looked like one big, messy, uniform cloud.
But the new, clearer analysis revealed what the paper calls a distinct bimodal distribution. Now, in simple terms, that just means that the messy cloud wasn't a single cloud at all. It was actually two totally separate groups that had just been overlapping and hiding each other. And this is a huge deal. It suggests that what we all thought was one type of EV is actually at least two distinct types. One group, which has a lot of a protein called CD9, seems to come right off the cell's outer surface. But the other group, with high levels of a different protein, CD63, probably comes from deeper inside the cell. This could mean they have completely different origins and maybe entirely different jobs in the body.
So, what does this all mean for us, for you and me? Well, this highly technical breakthrough actually has massive real-world implications.
I mean, why does all this matter? Because being able to accurately find and classify these rare EVs, it's a total game changer for medicine. This tool could pave the way for discovering brand new biomarkers, maybe leading to simple blood tests that detect diseases earlier than ever. It deepens our basic understanding of how our bodies work, and it could even help us design new therapies that use these vesicles to deliver drugs exactly where they need to go.
For decades, our cells have been broadcasting countless messages on a network we could barely access. With tools like this, we're finally starting to tune in properly. And you have to wonder, the question isn't if we can hear them anymore. It's what other profound secrets are they about to tell us?